Right, so today's lecture on bacterial genome dynamics and evolution, and we'll have to rush through because we're 10 minutes late. So what I'm going to do is just quickly remind you about what bacterial genomes look like, look at the kind of changes that go on in bacterial genomes, how they evolve, look at some case studies in more detail, and particularly how kind of counterintuitively sometimes to become virulent organisms lose genes rather than gain them. And also just look at the kinds of microevolution we see. So this is a figure from a review I wrote in Nature a few years ago, just looking at the three main forces, if you like, that are sculpting bacterial genomes. So we've got gene loss, gene gain, and gene change as the three things going on. And I'm going to go through some of these in more detail, in fact most of these in more detail during the rest of the talk. This figure here, taken from a recent review by people from the Sanger and elsewhere, also summarises some of the same key points. So you have mutation, you have gene duplication, recombination, gene loss, gene acquisition, and various kind of ways in which you can look at population genomics. And we'll say more about what we mean by pan-genomes and so forth later. In fact, on the very next slide. So just to remind you, what we mean by the core genome is the pool of genes shared by all the strains of the same bacterial species. So if we're looking at E. coli, we're looking at all the genes that ever reside within an E. coli cell of any kind. We then have this accessory or dispensable genome, which is the pool of genes that are present in some of the strains within that species, but not in all of them. We talk about the pan-genome to mean all of the genes which are present in the species in any kind of cell. Don't have to be in all of them, but in any one of them. And metagenome, the term is applied to the gene repertoire of a mixed population of cells. So kind of one stage further on. So you could talk about the gut microbiota metagenome to be the sequences associated with all the bacteria that live within your gut as a mixed microbial population. Illustrate this, these kind of concepts more concretely with E. coli. This figure here comes from a very early paper on the genomics of E. coli back in 2002. This is the first time where we had 3 different bacterial genomes from the same species from E. coli. And what we found, surprisingly, was that actually that core genome, the number of genes that were present in all 3 strains, is actually very much a minority of the pan-genome, of all the genes present in the species. Is that under 40% of the genes were present in all 3 of those strains. And each strain had its own complement of strain-specific genes, and then there were some overlaps between strains as well. So you can see it all illustrated here in the Venn diagram. But out of all the proteins, we have 7,600 and something proteins forming the pan-genome. The core genome was only a fraction of that, just under 3,000 genes. If we look at that another way, we can kind of imagine the E. coli core genome here along the axis here, forming that horizontal line, and then these vertical lines coming off of it are strain-specific gene clusters that are kind of, if you like, wedged into that core genome as you go along the genome. So the genome above there, CFT073, is from a strain of E. coli that causes urinary tract infection. The one underneath is a strain that causes diarrhea. It causes actually enterohemorrhagic infection. And you can see all the way along there are these wedges of genes. They're not kind of randomly distributed, though. They do appear to be certain hotspots along the genome where you acquire new genes. Now, beneath that is a figure produced from our own resource, XSpace, looking at comparisons between E. coli genomes. And in the center there's K12, which is the common laboratory lab strain, the common commensal lab strain that is used in molecular biology. And there it's been coloured in just by GC content. but the genomes around it from various different kinds of E. coli have been coloured in, where green is the stuff that's conserved with K12, and these little red wedges of red show you genes that are specific to that particular strain in a comparison with K12. So you can see in every case there are bits of the genome which are different, where there are strain-specific genes separate from K12. So this idea of genomic diversity has actually borne out and actually flourished as a concept the more genomes we look at. And in fact, you can take, now we're getting to the stage where we have dozens and dozens, even hundreds of genomes, but even at the stage of dozens of genomes from the same species, you can start to plot what's happening to the core genome and what's happening to the pan-genome as you expand the number of genomes you have. So what happens here is that this core genome has shrunk. So instead of, when we were looking at 3 genomes, there were about 3,000 genes. We said those are the core. As we've gone on and sequenced more and more, it's actually trended down now to around 2,000. And we're kind of probably reaching a stable point where we can say, well, actually, it's probably not going to change that much. All E. coli's have 2,000 genes in common in their core genome. If we look at the pan-genome, we can see that this is actually just keeps going up. There are various ways in which you can calculate what is the pan-genome and what is the core genome. Depends on what criteria to take to say that this is the same gene in 2 different strains. They may not be absolutely identical. You might say, well, they're the same gene. They're 98% identical in terms of nucleotide sequence. They are the same gene, effectively. Whatever criteria, and if you look at things like insertion sequences that jump around the genome, to say that this is exactly the same, equivalent in one strain as in another strain, it's in a different site, well, you know, it's flexibilities. So what you see here are 3 different measures, but you can see in each case the number of genes in the pan-genome is actually going up quite substantially, and we haven't yet saturated that pan-genome. So we can reliably predict if we go and sequence another E. coli that we've never seen before, we will find some new genes in the E. coli pan-genome that we've not seen before. So how do we gain genes? Well, one option is to duplicate genes. And gene duplication occurs fairly commonly. When this happens, you get 2 copies of the same gene in the genome. We create what we call paralogs. So they are homologs. They have a common ancestor. They show sequence similarity that indicates they have a common ancestor, but they're not in different genomes. They're actually in the same genome, and that's what we call paralogs. If they were in different genomes, we were looking at the equivalent gene in one E. coli and another E. coli, those would be homologs, we'd call those orthologs. But where it's 2 genes in the same genome that have arisen through gene duplication, that's what we call paralogs. And in fact, depending on how you set your cutoffs for really how similar things have to be considered homologs or paralogs, actually a very large fraction of the genome consists of paralogous genes. Somewhere between 30 to 55% of all the genes in a given genome are going to be paralogous. And there are large gene families out there. If we look at, say, flagellum, bacterial flagellum, we can see that actually the components of the bacterial flagellum have arisen through a process of gene duplication and then diversification. So it's not just that you get 2 things that are identical. They start to march off in different directions. They acquire slightly different sequences, then start to acquire different functions, similar functions, but just subtly different until they actually diversify enough to have different roles. You see this kind of thing in all sorts of settings. So if you look at, say, transporters that are transporting substrates in, you might have one that transports in glucose, it gets duplicated, and then it might wander off and start actually favouring maltose or some other substrate. And this is a common theme in evolution, both in bacteria and in other organisms, but particularly in bacteria. Another way you can gain genes is through horizontal gene transfer. So instead of the idea that lineages just march along from parent to progeny, passing on their gene sets in an orderly fashion with maybe a little bit of a change in a kind of clock-like fashion as you go along. in bacteria, and that's kind of what you'd see in mammals, let's say, us and chimpanzees and gorillas and whatever. But if you look at bacteria, what you see is actually very large degree of horizontal gene transfer, where genes are actually shuffling across sideways from one cell to another cell, not in a parent-progeny relationship, but actually just two quite separate cells, and genes move across. And this we call horizontal or lateral gene transfer. And one of the big surprises of the last couple of decades is the extent of this in bacteria. In a sense, you can argue that the whole bacterial world is involved in some kind of great orgy of genetic recombination of bacterial sex, swapping genes all over the place. Really quite prolific. And this is happening through a variety of routes, phages, plasmids, pathogens. I'll say more about that later. But it accounts for huge genomic diversity within a single species. So as we saw earlier, the core genome of E. coli is only those 2,000 genes, but you might have twice as many, or even, the largest E. coli genomes might have 5,000 genes in them. So 3,000 of those genes are moving around through horizontal gene transfer. Only 2,000 are actually going through the normal pathways of vertical descent. We tend to make a distinction between those things. Some people call it the mobilome. I think that's a neologism too far. It's a bit of a play on words, but they talk about the mobilome, this mobile part of the genome. Those genes tend to be niche specific. So if we say all E. colis, all E. colis need to have a ribosome, all E. colis need to have RNA polymerase, and so forth. And all E. colis need to make components of their cell wall in the same way. But some E. colis might be adapted to one particular niche, the ones that want to cause urinary tract infections, let's say, or the ones that cause infection in the gut or invade and so forth. And those niche-specific genes are what we find in this horizontal component of the genome. Just to remind you again of the kinds of elements we're talking about here, we talk about mobile genetic elements that are actually involved in this horizontal gene transfer. We have transposons, bits of DNA that can jump around, so jumping genes. Some of those, there are more sophisticated versions in bacteria which are called conjugative transposons. So they don't just jump around within a cell, within a genome, they actually encode mechanisms to allow them to pass from one cell to another through a process of conjugation. So they'll get two bacterial cells to come close together, and then they'll push their own DNA through from one cell to another. Insertion sequences are in the other direction. These are the most cut down version of a mobile element you can have. All they do is they have a gene that encodes a transposase, and they just jump around the transposase, and the flanking regions just jump around. You might say, well, that's a kind of trivial thing, but in fact they do play a role in kind of the genome architecture, because as they jump around and go into different parts of the genome, they provide then a template for homologous recombination. So the cell has this mechanism for recombining pieces of DNA that are identical or nearly identical, and in this way bits of the chromosome can get shuffled around through recombination, through insertion sequences. Say more about that in a moment. Plasmids, we've spoken about before. These are usually circular replicons. Bacteriophages, bacterial viruses that can carry DNA from one cell to another. Another fairly sophisticated kind of mobile genetic element is the integron, where here you have these kind of these cassettes of genes that can be shuffled into in front of a promoter in various kind of versatile ways, and you end up with these whole large cassettes of things moving across very en masse between one replicon and another, and typically with antibiotic resistance you get a whole load of antibiotic resistance genes all stacked up together and moving as one unit through these integrons. Another key feature of genome evolution are these so-called genomic islands. Otherwise, these are a generic example of what are sometimes called pathogenicity islands. They're large chromosomal regions. They move in ways which are fairly mysterious, and sometimes they have integrases associated with them, sometimes they don't, and we don't quite know how they've been mobilized. But you can look at one strain that has a particular phenotype, causes diarrhea, and look at a strain that doesn't, and you'll see somewhere in that genome wedged in will be a large cluster of genes that might account for that increase, that change in virulence potential. Here in cartoon form, it shows you the sorts of things you might see. You might see an integrase, you often see tRNA genes where they've been shoved in, direct repeats flanking it, and a range of different genes in there, a cluster of genes carrying into the genome a certain functionality. Some people talk about these genomic islands giving the organism a quantum leap up to a new phenotype. So instead of an organism having to slowly work out how to colonize a particular niche by changing one gene after another, slowly, stepwise fashion, it just acquires the whole shebang in one go through having a genomic island enter the genome. This is taken from a review article. I've got a number of papers I need to put up into WebCT for sort of further reading. This is one of them. I will do that in the next day or so. This just gives you a feel for how widespread these kind of genomic islands are and the kinds of functions that they encode. So things, everything from nitrogen fixation, resistance to antibiotics, production of toxins, fimbriae, all sorts of things. Okay, so that's gene gain. What about gene loss? So if you think about it, if you've got a bacterial genome, and we have this steady rain of genes coming into that genome through horizontal gene transfer, if things stayed as they were, if that was all there was to it, the genome would just get bigger and bigger and bigger and bigger. But actually, bacterial genomes stay roughly the same size. They are kind of, they're a few megabases, no more. And so, logically, we have to accept that gene gain is balanced by gene loss in the long run. If you compare, say, E. coli and Salmonella, they have roughly the same genome size. They've gained lots of genes, but they've also lost lots of genes along the way from their divergence from a common ancestor. Why do you lose genes? Well, sometimes if the phenotype they're encoding is no longer needed, in fact, it may actually be producing a cost in that new environment. For example, producing a bacterial flagellum is quite costly. If you're in an environment where you don't need to be motile, never need to be motile, then just losing the flagellum actually is a good thing. So just knocking out those genes to start with and then losing them is going to be useful for you. If you actually just knock out the gene function so that the genes are no longer producing proteins, but they're sitting there in the genome, there is a marginal fitness cost in just producing, you've got 20 kilobases of DNA that's not doing anything in the genome. There is perhaps a marginal cost. But that's a slow thing. So the first thing that happens is actually you get introduction of stop codons into a gene, turning it into a pseudogene. So what we mean by a pseudogene is a protein coding gene that no longer is capable of producing a functional protein. So if you stick a stop codon halfway into a gene, halfway through a gene, or you frame shift the gene, you might get a little bit of the peptide produced, but it won't be a functional protein anymore. Then slowly what happens is that those bits of sequence will start to accumulate more mutations. In fact, it's not shown in this slide here, but you often get insertion sequences jumping into those, and those disrupt the genes, and they provide a substrate for homologous recombination, and eventually the gene will get snipped out of the genome. I'll give you an example in more detail from our own research from a few years ago. We were very interested in a cluster called ETT2, which is found in E. coli 157. You can see that this whole genomic island is present as a 27.5 kb insertion relative to some of the lineages like the UPEC and EPEC1 lineages that have never seen this cluster. And if you look in E. coli K12, what you find though, I mean, we kind of tend to think in shorthand terms that E. coli K12 is like the archetype of all E. colis. It's not. It's just a lab strain, but by kind of chapter of accidents happened to be the one that most people work on. But here, E. coli K12 is not looking like the ancestral state at all. It's actually had this island jump in, but then a large part of it, over half of the island, has just then been lost from the genome. So if we look at the GC content in 0157 for this region, we can see the GC content swings down to be very low for this region here. And then there's a fraction of this region present in K12. But tellingly, just at one end here, there's some insertion sequences, and that is like a smoking gun. And what's probably happened is this ETG2 actually produces a type 3 secretion system. We don't know what the function of that secretion system is, but one assumes it's no longer needed in this lineage. So the genes are no longer, they're silenced by point mutations and so forth. And then the whole region starts to get degraded as insertion sequences jump in at two different ends and then provide the matrix, if you like, for recombination to occur and that piece of DNA to be snipped out. And in fact, we kind of have a Russian doll effect here. If we look at a whole range of different E. coli lineages, we see progressively larger deletions in this region. So in 0157, 042, we have the whole gene set there. In EPEC2, we've got an 8 kb deletion. In K12, 14.6. In Shigella sonnei, we have a larger region deleted, most of it, 3/4 or more. And in Shigella flexneri, which is another kind of E. coli, although we call it Shigella, it's actually genomically belongs within E. coli, there's just a few hundred base pairs left of this island, and loads of insertion sequences at that locus where the island was. So this is the way in which genes are lost from genomes. They slowly decay away like this. And just as an example to show it's not a one-off, I mean, we actually came across another locus very similar, which we called FLAG2, a second flagellar system in E. coli K12, totally unexpected. But it turns out if you look in E. coli K12, there are these two flagella genes by homology, but they just sit there on their own, not doing anything very much. But if you look in other E. colis, like E. coli 42, you find at that locus there is this whole, this point in the genome, there's this whole locus of 40-odd genes encoding a flagella system. So again, this is an example where islands go into a genome, and then they get, if they're useful, they get retained, but if they're not useful, then they get slowly degraded away. And some people use the term palimpsest to describe a genome. So palimpsest is an ancient medieval manuscript where, when parchment was very expensive, someone might write something on a parchment and then they would scratch it off and write something else on top and do this several times. And it's a bit like that. The genome is not a fixed entity. It's been kind of written on, and then scratched away, and written on and scratched away over time. Another important concept, slightly counterintuitive, is the idea of pathoadaptation. So you can imagine, okay, so to become a pathogen, you're a bit like K12, you're kind of a boring old E. coli, and then you get a wedge of genes come in, and that, bang, you suddenly become a pathogen. And we can argue that to evolve Shigella, which is a specific kind of E. coli, we'll say more about that later in the course as to how it causes disease, what we're doing is adding genes. We're adding a virulence plasmid. We're adding these pathogenicity islands, and these turn a common or garden harmless E. coli into a nasty pathogen. But there are also, this pathogen also illustrates the fact that you can lose genes along that route. And in fact, not only can you, but you have to lose genes to actually get to the fittest state of being a new pathogen. So Shigella, when it became a pathogen, started living inside eukaryotic cells. And one of the things that's seen in, and in fact there are several lineages of Shigella which all appear to have evolved towards being a pathogen separately. So they've all acquired these virulence plasmids, but it seems to be a separate event. And if you look in their genome, this is not coming out particularly clearly, but if you look in the chromosome, you see that they've all lost the function of one particular gene, CAD-A, which encodes this enzyme lysine decarboxylase, which produces this molecule here, cadaverine. And the fact that this has happened in all of these different lineages, and the fact if you stick cadaverine back in again into Shigella, you see changes in virulence, suggests that this is adaptation to virulence. It's a loss of a gene which, if that gene was switched on, would actually hinder survival in that virulence niche. And in fact, there are others. There's loss of flagella motility. So Shigella are always non-motor. They've thrown away the flagellum. They've lost this outer membrane protein OMP-T. We still don't quite know why you have this loss of genes, exactly what's going on. This is from a paper where they tried to fathom this out, and they basically they showed that the wild type, which doesn't have cadaverine, and the cadaverine had A positive things, they had various things that were just the same. So the Shigella cells could invade HeLa cells just as well, they could form plaques just as well, they could induce apoptosis just as well. But in terms of if you put Shigella into an ideal loop, you can do this ideal loop assay, you get fluid accumulation. That's what happens with the wild type, but it doesn't happen when you have that cadaverine gene on. And similarly, you've got differences in the migration of polymorphs in there. And so people are still teasing out exactly what's going on here, but it's an example of gene loss being important. Here's another example of pathoadaptation where you get gene loss and gene gain. This is a paper from a friend of mine that looked at the interaction between LPS and a type 3 secretion system that was present in Shigella. So to get to the stage of being able to invade cells, Shigella has to have this type 3 secretion system. But these cells get this vigorous, it induces vigorous inflammatory response, and they will get killed if they're just as they are. So they produce this LPS, which it sticks out from the cell. It's a long extended molecule. And if they actually produce the LPS and the type 3 secretion system, they reduce the resistance to killing is okay, but they then can't deploy the type 3 secretion system because the LPS gets in the way. So what they've done is they actually create a variant form of the lipopolysaccharide, which is shorter, closer to the surface, allows the type 3 secretion system to be deployed, but also provides the resistance to killing. So this is a kind of complex way in which these two different features of the cell surface interact to give us a virulence phenotype, which is distinctive and different from the ancestral state. Another interesting thing we see when we look at genome evolution in pathogens and in bacteria in general is what happens when a bacterial lineage shifts from one niche to another. So typically when a generalist kind of organism that can live in lots of different environments suddenly starts living in one rather boring, predictable environment. What we see when this happens is we see the organism suddenly has to jettison large parts of its genome, because large parts of its genome are either no use to it at all and will get just lost slowly but surely, or in fact they may be a positive hindrance and get selected against. And we see this as a very common kind of motif in bacterial genome evolution. There are a number of important pathogens of humans which are where you have this kind of population bottleneck. You get this very genetically uniform clone of an organism moving into a new niche and then being cut off from the rest of the population, becoming quite specialized, and then slowly, well, not slowly, quite dramatically, throwing away most of its genome. So here are three examples. Mycobacterium leprae, which lives only on humans, causes leprosy, lives only inside our cells. It has thrown away a huge amount of its genome. And we can see this in progress. Over a quarter of the genome of Mycobacterium leprae, the genes there are present in the genome, but they're not functional. They're disrupted by frame shifts and stop codons. They do not work. They don't make proteins. Even though you can still see most of that protein coding gene there, you can identify it and say, oh, that is an ortholog of something in another mycobacterium that has a function we can assign and so forth. They just don't work. We see similar things in Salmonella and in Shigella. I mentioned Shigella before. In Shigella, we get 11% of the genome is pseudogenes, genes that have been switched off because they're no longer needed in the new environment. And similarly, Salmonella typhi is another example of a very, what we call monomorphic pathogen, where actually the population structure is very constrained, there's lack of diversity in that population, and that population is evolving to become very specialised in a particular niche. So Salmonella typhi, again, only infects humans. Spreads from human to human through fecal-oral route, but it doesn't affect any animals, doesn't live out there in the environment, and it's become quite specialised. Similarly with Shigella. Here's another example, Bordetella pertussis. Is again a very specific pathogen of humans, doesn't cause disease in other animals, doesn't live in any environmental reservoir, it just spreads from one person to the next person, lives in their throat, very constrained environment. And when people first, in the early part of the millennium, people started looking at Bordetella genomics, they found that the Bordetella pertussis genome had not only just got lots of pseudogenes in it, it acquired lots of insertion sequences and had undergone extensive rearrangements compared to the ancestral state. So here you have a three-way comparison between Bordetella pertussis, a dedicated human pathogen, and two other related organisms that have a much wider host range, Parapertussis and Bronchoseptica. And you can see if you compare Bronchoseptica and pertussis, this here is the whole genome laid out in a line. These red bars here are showing parts of the genome that are collinear, that are the same in the same place as you look along the genome. And you can see between these two organisms that have a generalist lifestyle, it's all pretty much the same. There's been a part that's swung around in the middle, but pretty much collinear. But when you compare Bordetella pertussis with Bronchoseptica, you can see here the whole genome's just been randomized, if you like, shredded around, moved around. And this is a result of the fact that it's moved into a new niche, it doesn't need most of its genome, and there's a kind of proliferation of insertion sequences. So normally, insertion sequence can jump around in the genome, but if it tries to jump into an essential gene, then that cell just dies. If it jumps into a non-essential gene, then it can survive there. So when you get to a situation where a large part of the genome is no longer essential, or no longer even provides any selective advantage, then that provides a kind of fertile ground for these insertion sequences to proliferate, and that's what we see here. And then once they've proliferated in the genome, you then have multiple sites for homologous recombination to occur, and so the genome can get randomized like this, can get mixed up. How are we doing for time? Probably just about going to make it. Another case study let's just quickly look at is Yersinia pestis. So this is a bacterium that causes plague in humans. So there is a mammalian reservoir out there, lives in rodents, and then it spreads into other organisms, including humans, from those rodents. This organism also has to live in fleas, so it has to live in mammals and in fleas. And there are a number of gene products that actually help it live in fleas. There's this HMS gene product that makes the bacteria form aggregates. They block the foregut of the flea, and so the blood is regurgitated from the flea. And so the flea bites one host, takes in this blood meal, then bites another host and regurgitates the earlier blood meal, spreading the bacteria that are into it into the new host. Now, Yersinia pestis in humans and in other animals can actually live inside the circulation, inside cells of the immune system as well. It resists phagocytosis, it can kill monocytes, it can live causing septicemia. Now, this distinguishes it from its closest relatives, which actually are just gastrointestinal organisms. They live in the bowel of mammals, and they spread from one mammal to another, fecal-oral route. Occasionally they can invade. They're not particularly good at invading the tissues. Occasionally they can invade and cause disease, but they're nowhere near the same as the fearsome pathogen that causes plague. And so we have a conundrum as how do you get from one of those kind of organisms to this killer organism, Yersinia pestis? Well, looking at genomes, it's clear that Yersinia pestis evolved from Yersinia pseudotuberculosis. So Yersinia pseudotuberculosis lives in the gut. This is actually quite a varied species, and Yersinia pestis, if you like, is one particular lineage that actually specialised and changed. And how did it do that? Well, it acquired, how did it go from being an enteric pathogen to a flea-borne blood pathogen? It acquired these plasmids, one plasmid that encodes a plasmidogen activator, which allows it to survive and disseminate from the site of infection. Capsular antigen, which blocks phagocytosis, encoded on another pathogen, and a murine toxin needed to survive in the flea. And the ancestral Yersinia plasmid species already had one plasmid in there, which helped to overcome the host immune system. So we got those plasmids, then acquired pathogenicity islands and recruited various chromosomal genes to new functions. So we've got all these things listed here: type 3 secretion systems, insect viral enhancing factors. So there were a number of changes in the chromosome itself. We also got this massive rearrangement of the genome and proliferation of insertion sequences. So very much the same kind of changes we saw with Bordetella pertussis. So this picture here of the whole genome shows us regions of the genome that are inverted, switched around. So normally if we plot GC skew, it basically shows us the genome in two halves, diverging away from the origin of replication. Whereas here, in fact, what we're seeing is parts of these are actually flipping around. So this is because parts of the genome have moved in recent time, if you like. Lots and lots of insertion sequences, lots and lots of pseudogenes in the genome. Here's some of the pseudogenes that in ancestral Yersinia would have had interesting functions to do with virulence. We'd have annotated them as virulence associated. Flagellar genes, that's another issue. Flagellar motility lost here. Various cell surface proteins, various enzymes. All of these have been lost because they're no longer necessary in this new lifestyle. Okay, just a few minutes left, so we did start late. This basically takes us into, you can see where this is going, that basically if organisms actually get very specialised, they're going to start losing parts of their genome. Now, if they get cut off from the rest of the world, they start losing genome, but they have no way of gaining any new genes. And so they get cut down smaller and smaller and smaller. And this figure here just gives the kind of range of genome size and how it maps onto lifestyle. So very big genomes in free-living organisms, very few pseudogenes, a lot of horizontal gene transfer, fairly stable. You then move into these organisms like Yersinia pestis, Mycobacterium tuberculosis, Mycobacterium leprae. They become pathogens, become adapted to it. There's people waiting outside, so that stop soon, haven't we? They have some pseudogenes, but you know, they have actually a proliferation of pseudogenes and a proliferation of insertion sequences, and then eventually they get so cut off that they purge themselves of all the pseudogenes and all the insertion sequences. They don't have any more DNA coming in, but they get very small specialised genomes. And this is particularly common in intracellular niches. So organisms that actually live inside eukaryotic cells as their niche have very small genomes. And this is the alpha proteobacteria, one particular lineage, seems to do this a lot. We've seen a lot of rickettsias and bartonellas. They undergo this downsizing dramatically. Sometimes the downsizing, they still keep collinearity. So this is a plot of one genome against another, pretty collinear. In other cases, the genome gets completely shredded as well in the process. And the reductio ad absurdum, well, we get down to a genome of 160 kb, this organism Carsonella ruddii. It's an endosymbiont of psyllids, so we end up with endosymbionts rather than pathogens. And then if we go further still, we end up with mitochondria, which are actually bacteria, which have thrown away the vast majority of their genome. Much of it's actually been exported to the nuclear genome, but they end up with these very, very small genomes. Right, the last slide or two is just to talk about microevolution. So we're talking about the big picture up till now, about bits of genome being slung around, inverting, huge things coming in. But of course, genes in the genome are undergoing small-scale mutation. They're acquiring point mutations, they're acquiring little insertions, little frame shifts going on, deletions and so forth. And this is going on all the time. And this guy here, Moto Kimura, back in the 60s, actually recognized that if we look at what's going on at the genomic level, in biology, at the genetic level, there's this steady accumulation of mutations, but they're all neutral mutations. They're not making any difference to the phenotype, to the biology of the organism. You're just getting one codon that codes for glycine turning into another codon that codes for glycine. Or you're getting a codon that codes for glycine turning into one that codes for serine, but they're doing the same kind of thing in the protein, so it doesn't make any difference to the function of the protein. And so scientists now very commonly look to see, they look at the neutral background stuff, and then they look for changes where there may be adaptation going on, where actual natural selection is working and making a change in terms of not just the genotype, but the phenotype as well. And a common way of looking at that is to look at the ratio between synonymous and non-synonymous mutations. So if you think that the mutations are coming in all the time, clock-like fashion being accumulated, every gene will have a certain ratio of the ones that change amino acids to the ones that don't. But the ones that are actually moving fast because they're actually changing in response to the environment, to some adaptive change, they will actually show an excess of changes that make differences to amino acids over the ones that don't make differences. And so that's often used as a measure of which parts of the genome are going on. Not getting too much pressure from those people out there. We'll continue for a few seconds more. So we get these SNPs, we get these small indels. We can use this as a clock, if you like, to actually draw up, we can get diversification, we can draw up phylogenetic trees and follow evolution of strains. But the problem is that in some bacteria, many bacteria, they're actually exchanging DNA with their partners. So you might have two lineages come close together and they exchange a slab of the chromosome. 5% of the chromosome suddenly goes from that one to that one. 10% of the chromosome goes from that one to that one. So if we try and draw family trees based on all the genes in the genome and the changes in them, we don't get clear pictures. And so for these organisms that we say are highly recombinogenic, things like some salmonella, well, Salmonella, you can see here that some of these are showing a kind of tree-like branching, but if we look at things like Neisseria meningitidis and we zoom in, we can't draw trees. We kind of see these starbursty kind of patterns, and we talk about clonal clusters and sequence types rather than lineages in the phylogenetic sense. Recombination, let's move on quickly. I think I'll skip that as well, and I think I'll finish there. So we've taken you through the nature of bacterial genomes, the ways in which they change, gain, loss, accumulation of pseudogenes, downsizing in isolated niches, and pathoadaptation. Good, thank you very much.